The next speaker is Professor and Neurologist and the Director of the Kids Neuroscience Centre at Westmead, partly Sydney Children's Hospital Network and University of Sydney. And his expertise is in a really hot area of science at the moment, in immune dysregulation. I'd like to introduce Professor Russell Dale.
Thanks, Adam. So if you're going to take away a few things from my talk, first is many genetic and epigenetic conditions, they don't just affect the brain, they affect other organs, including the immune system. And our immune system and our brain is constantly interacting. So those are probably two of the big take-home messages of today, and this is just some detail. This is a sort of spectrum of neurodevelopmental disorders from an etiological perspective, monogenic conditions and then more gene environment. And, 10% of children have a neurodevelopmental disorder, and many of them are not monogenic. They're sort of in the more vulnerability gene plus epigenetic space. And this is something that isn't necessarily thought about much, that the immune system is pretty central to brain function. And here's just a few sort of comments. First is that a lot of kids get these setbacks, and I don't know whether that is a big feature of Prader-Willi. I don't claim to be an expert in Prader-Willi syndrome, but it's certainly a big feature of autism and neurodevelopment in general. The children often have setbacks or regression, and our approach to it as clinicians is poor. We just acknowledge it exists. We don't have a framework or a clinical therapeutic approach to it. The second thing that you can hopefully see in the middle there is that brain development is very dynamic. That makes sense. We know that. We see a baby and then we see an adult. Of course, the brain develops, but it's extremely dynamic synaptic connectivity, but also removal of synaptic connections. The other fact is that 10% of our brain cells are actually immune cells. So we have an immune system in our brain. It's called the microglia. And the microglia do most of the synaptic pruning. So they're constantly removing synapses that are not being used. And so we can end up with too many connections or too few connections, and this is pathological. And then the final point is that the immune system in the brain is constantly interacting. And also, of course, gut health. I'm not really going to talk about the gut, but gut, immune, brain is an interaction that goes on. So this is a hypothesis that we've been running with, that gene plus environment, i.e. epigenetic, immune brain dysregulation is a feature of neurodevelopmental disorders. And if you want to measure epigenetic factors or gene regulation, then RNA sequencing is a very useful way of investigating this. A first way to sort of think about immune problems is, do children with neurodevelopmental disorders have immune problems? And a lot of children with autism and neurodevelopmental disease have higher rates of asthma, allergy, immunological problems, but also recurrent infection. But we're not very good at measuring just normal infection like throat infection, ear infection. Pediatricians usually good at screening for immune deficiency, but not for recurrent infection. We just sort of dismiss it, or recurrent throat infections. But this is a study we did. These are kids with neurodevelopmental diseases here, and these are controls. And this is in the first 5 years of life. And this red means more, essentially, and white means less. And so you can clearly see here that this is family reports that children get recurrent infections, particularly throat and ear infections that you can see here, but also mouth ulceration and needing to go to the GP. So you could argue, they're just over-reporting this, but actually this has been shown at a data linkage at a population level that children with neurodevelopmental disorders get more infections. And also they get quite a large amount of setbacks or regression across all domains there: social, attention, cognitive, and so forth. We've investigated a particular clinical syndrome, which is a controversial syndrome. It's something I personally believe in because I've seen too many families and children that have this phenotype. But the way I think about it is this is a clinical phenotype. rather than a magical disease. This is what has been called pediatric acute neuropsychiatric syndrome. Now, you can just tell that's a bit non-specific, isn't it? But what it's describing is that children present very abruptly. These families often present the kids to emergency departments because they say, my child has changed. They often get acute onset OCD. And it's often infection provoked, but we don't really understand it. Is this part of just normal neurodevelopment, or is it some kind of separate entity? And I think it's not clear yet. But the entity exists, but what is it is not clear. So what we did was we did some RNA sequencing, and first thing to say, this is just a bioinformatic approach, but what it does is it separates you can see here the patients with PANS separate nicely from the controls in the study. That suggests there's a difference. It doesn't tell us anything more than that. And we can use the RNA. What happens is it measures all of the RNA expression, all the different reads of RNA in patients compared to controls, and then uses that and inputs that into pathway analysis. And the upregulated pathways are in red and the downregulated pathways are in blue. I don't want to spend too much describing this, but essentially most of the upregulated pathways are ribosomal pathways. Ribosomes are what turns RNA into protein. So it's as though the ribosome is being overstimulated. And these are the genes that were part of these pathways, and most of these are ribosomal protein that are upregulated. And here's examples here on the left-hand side in the patients versus controls. So you can see fairly convincingly that these ribosomal proteins are elevated in these patients compared to controls. We also found methyl transferases. Certainly that's an interesting name of genes for this audience. So transferring methyl groups to RNA in this case. So they were also upregulated in these patients, suggesting some epigenetic phenomena occurring. What about the downregulated pathways? Whoops. The downregulated pathways in blue, these were mostly immune pathways. You can see here neutrophil degranulation at the bottom there. These are, so it seems as though there's a reduced immune pathways in these patients. And these genes were a whole array of toll-like receptors, complement, and other immune function genes. And again, there's examples on the right-hand side of these genes are decreased in the patients compared to control, in contrast to the ribosomal proteins. And so if you summarize this, if you're talking about epigenetics, what we mean is gene regulation. An epigenetic control can occur at a DNA level, like DNA methylation, at an RNA level, so modification of RNA, but also at a protein level, protein modification. But if you summarize our findings, there are methyl transferases are elevated. There's a problem with translation, which is ribosomal function in production of protein. So what about the function of the immune cells? We've said that they have recurrent infection, these children. So we did a functional immune assay called a toll-like receptor assay, which is basically, we use lipopolysaccharide, which is a bacterial substrate to simulate immune cells. And the pink is the patients versus the green is the controls. And you can see this is for interleukin-6. There is a reduced response at 3 hours, 30 minutes and 3 hours in patients compared to controls at an RNA and a protein level, suggesting indeed there is a form of immune deficiency. And really, this is the so what slide. And I think I prescribe guanfacine quite readily, and it definitely helps a lot of kids. So do SSRIs, so does risperidone, so does aripiprazole, so does psychology, so does therapy, but it doesn't always help all patients all of the time. And there is this mantra in psychiatry that about 30% of patients are refractory. About 70% of patients respond to conventional treatment, but about 30% will be inadequately responsive. So what next? And I suppose the big question is, if we're talking about precision medicine, you want to target upstream causes rather than downstream effects. That's a sort of model of medicine, preferably. So totally relevant to this audience, epigenetic therapies. If we're talking about epigenetic therapies, what do we mean? Well, there's a lot of fairly cheap and safe things like butyrate. Butyrate is produced when you are in ketosis. It's the ketogenic diet. The active metabolite of the ketogenic diet is butyrate. And it is an epigenetic metabolite. It's an HDAC inhibitor, if that's of interest to you. So we could potentially have drugs that can modify the immune system or the epigenome. So what about Prader-Willi? Well, how might we approach this if we were to frame it in an immune brain way? Well, the first thing to say is the genetic defect expressed in immune cells as well as brain cells? Some genetic conditions are brain specific. The majority are not. The majority affect brain, immune, heart, and other organs. So is there a genetic immune defect that results in immune dysfunction in Prader-Willi syndrome? Second question, do people with Prader-Willi get recurrent infection or immune problems? Are they prone to setbacks? What about RNA sequencing in Prader-Willi syndrome? Has it been done? Will it inform us about cell function? What about immune function, and will this result in new therapeutics? And this is my last slide, really, that I think the actual animal model and the biological understanding of neuroscience is about 20 years ahead of clinical care. And if we want to learn in neuroscience about novel drugs or the drugs that we'll use in 20 years' time, which speciality should we look at? Oncology. We'll be using those drugs in 20 years' time. You can have, and they're doing amazing things, the sort of things they're doing now, and it's all precision medicine. So where hopefully in the future we'll be framing things that's an immune as well as a brain problem, neurodevelopment, and that it isn't just about DNA variation, it's all about gene-environment interactions as well, and that really therapeutics, we want to target disease mechanisms if possible. Thanks very much.
Is it on? Yep, very nice presentation. Maybe we should have a chat, because what you just said on the last slide, we've done all of it. So we have the data. We will have the data. So come and talk to us.
That's what we want.
No one else? I might just ask one question, Russell. Just whenever we hear immune marker type presentations, there's always a conflation between causal and correlates. Can you speak to how you sort of understand what causes versus what correlates when it comes to immune markers?
Cause and effect kind of thing. Yeah, well, that's just a fair point. I think the most important thing to do is ask that question and accept that might be a possible, in other words, confounders. The role of the immune system, if you look at the pathology of autism and the brain transcriptome data, they found inflammation for 20 years, but it's been quietly, don't know what to do with it. Instead, they focused on the downregulated transcriptomics around synaptic and neuronal function, and then microglial activation, which is the immune activation, is sort of ignored. And I think that just speaks to the fact that we're not quite sure about these points. The cause and effect, is this direct cause or secondary, primary versus secondary? No, these are all big questions. And, unpicking it with animal models, of course, are challenges. We can't measure the brain transcriptome easily, obviously. But if there were environmental factors, and this speaks to Lauren's point about the autonomic nervous system, environmental stressors and factors, they will not selectively affect your brain. Why would they? They will affect your whole body. So really, the blood may be a marker of dysfunction of the brain. That doesn't mean it's the same. But, our brain, our gut, our autonomic nervous system, our central nervous system is under the same environmental pressures of what we eat, our psychological burden, our stress, our fatigue, and all of those factors that constitute our environment.
I don't doubt a lot of what you say, but people with autism. Tanya Markovic, Sydney. People with autism, with severe autism, who are, you know from diagnosis that they've got it from soon after birth. So where does the immune system fit in there?
Look, I think my experience of neurodevelopment is there's this whole massive spectrum, and I know the patients you mean, but I've also seen the patients that genuinely seem pretty normal at the age of 2 and 3, and at the age of 5, they're definitely not normal. They truly regress.
So that's a different
I think
situation.
so. Well, I mean, we accept that neurodevelopment is an absolute spectrum where you have your genuine monogenic, your Rett patients who are clearly presenting at 6, 12 months of age. But then we've also got this spectrum of high-functioning autism, Tourette syndrome, ADHD, OCD, where the yield of genetic testing is low. I think you're referring predominantly to the more severe, much more likely to be monogenic, intellectual disability, severe autism group. But the spectrum of neurodevelopment is far more broad than that.
Yeah, I mean, these people aren't monogenic. They're not people with Prader-Willi. Yeah, they're not. It's different. Anyway,
look,
it's interesting.
I think.
So
my experience of neurodevelopment is it's extremely broad, and you're describing, a group,
so in some people
but not
it's
all.
more markedly genetic, and in others it's environmental.
No, a mixture of gene plus environment.
Yeah, that's right. Yeah. Thanks.
Yeah.
Sorry, Kathleen, we've got one more.
Hi, this is a little bit off topic, but it's related. Trauma. Dealing with what we know about trauma in early childhood. My child and children have come from trauma, and one has Prader-Willi syndrome and the other one has ASD, ADHD, intellectual impairment. Is there any... Their masks such similar, there's such an overlap. What causes what? What's related to what? Do you feel like we're getting any closer to kind of getting a direction so that we can try and intervene and help, but without having what is it related to? Is it normal development? Is it the fact that they're on the spectrum? Is it environmental? And what part does trauma play in their lifespan, whether that be from 0 to 2, whether that be at school? We all know that kids that are neurodivergent do struggle in our school systems, and later on in adult life.
Look, you're making an excellent point. I'm not going to give you a very clear answer because it's so big and the scope is huge. But trauma leaves its mark, and it leaves its mark epigenetically, and it changes immune function. And post-traumatic stress disorder, you just Google that now or do a PubMed, but post-traumatic stress disorder in immunology or inflammation, and away you go. It's because stress affects your immune function. We sort of know that, don't we? Yeah, so, but unpicking this, I think as a neurologist, the last 10 to 15 years have been amazing, that our ability to sequence the DNA has been transformative. But that's where we are, really. But what about RNA? What about protein? What about cellular function? We're not there yet, but why wouldn't that be part of this story? The future is we'll walk in 50 years' time, we'll get a drop of blood and they'll do high throughput DNA, RNA, protein, immune function, and spit something out. You have a 72% chance of X, and I'm not sure I want that. Yeah.
Thanks, Russell. Take your place. Okay. Look, no doubt it's a really hot topic, encouraging huge amounts of debate, and Russell's at the forefront of that science.